Greetings. Today I'll be discussing aging skin, reversing cellular senescence. So how do we age? You may be familiar with this diagram. These are the 9 primary hallmarks of aging. They represent basic health maintenance mechanisms which over time accumulate damage, are injured, or even fail, and it results in premature aging of cells, tissues, and organs. The good news, however, is that these are also potential points of therapeutic intervention. So there's some other exciting developments. Professor Lorna Harries and some others have worked on this issue of dysregulated RNA processing, and it appears to be a novel driver of cellular aging and may represent the 10th hallmark. And we'll discuss this a bit more later. So if this is how we age, how do we treat or even reverse aging? So these points of intervention are our targets, but they're not all equally weighted. And at this point, research is emerging, everything is evolving, it's very exciting, but we really have many, many things we don't understand. So we know that these are not all equally weighted. Even with identical twins, you might have a different lifestyle, different environment. There are genetic variations, different ages of your life. There are many, many issues. But generally speaking, very broadly, some of these may be more important early on, and as we develop therapeutics, that may become more important. So, for example, DNA damage, epigenetics, splicing may have some important roles in prejuvenation or earlier changes. Senescence, loss of proteostasis, and particularly mitochondrial dysfunction, and eventually stem cell exhaustion, those are critical issues later. Some of these may have threshold effects too, for example, DNA damage. So we have much to learn, but it's an exciting field. So let's discuss cellular senescence. So aging, injured, or damaged cells basically follow 2 main pathways. They either undergo apoptosis, which we're familiar with, the cells die and exit the organ, or they undergo senescence, which the cells become dysfunctional, but they do not die and remain in the organ. Cancer cells, unlike the senescent cells, which don't divide, cancer cells, as we know, have unregulated cell growth and unfortunately do not undergo apoptosis, and so also, like senescent cells, remain in the tissues and organs. So what do senescent cells do? Well, they're alive, but they lose their ability to grow, multiply, and divide. However, they refuse to die. They are these so-called zombie cells. They have altered function, which causes them to secrete these chemical secretory signals termed senescent associated secretory phenotype, or SASP. SASP are very critical. They are pro-inflammatory, and they have a variety of other potentially or very negative impacts. Really importantly, they also create a bystander effect. So these SASPs secreted by the senescent cells have a bystander effect on the surrounding normal cells, producing dysfunction in the normal cells. This then becomes a multiplier effect, and basically SASP appear to be some of the major drivers of age-related diseases. So how do these impact aging? Well, one issue to consider is since they don't divide, it also impacts replacement of lost cells, and this also can affect stem cell division. So there are a great number of studies in mice and limited studies in human at this point. Interestingly, adding senescent cells to younger, healthier cells induces aging. So it creates in mice a decline of physical parameters, strength, function. It induces senescent cells in the surrounding cells. There's an inflammatory increase and degradation of cellular protein synthesis. All of these indicate that senescent cells do impact the surrounding tissue. In contrast, if you remove senescent cells, and there's some old mice studies that show this, you can actually see improved function, cardiac, pulmonary, other issues that are important for perhaps not our skin and aesthetics directly, but they affect our body. You have improved muscle strength, decreased osteoporosis, and most of these mouse studies show increased lifespan, and they can be pretty dramatic amounts. Unfortunately, as humans, we have not seen these transfer over to the studies yet. So very hopeful, but we need more human studies. So to return to the cell pathways, so the cell pathways of apoptosis versus senescence have important differences. Apoptotic cells, as we know, they undergo programmed cell death, which is a natural process, and it eventually leads to the death and removal of the cells. However, senescent cells, in contrast, while they're no longer able to divide, they are still metabolically active. So they do not undergo apoptosis, they do not die, and they remain in the tissue and organ. So this bystander effect that we mentioned earlier, it affects their own cell removal rate. And if you think about this, as the senescent cell burden grows and the SASP bystander effect grows, it creates this snowball effect that actually can accelerate aging process. So we do not want these cells in any part of our body if possible. So how does this affect treatment? Well, there are two main treatment therapeutic pathways: senolytic agents and xenomorphics. Senolytic agents are the most popular, the most studied. That's where most of the mouse data is, and that's where some of the really exciting human data is. The senolytic agents remove the senescent cells. They die, they're removed from the tissue, and that stops the SASP, so it allows the non-senescent surrounding cells to function more normally. It does deplete the organ of the total number of cells, which can help improve organ function. There are some concerns if the organ has mostly senescent cells, that perhaps this actually stresses the organ and can, in the long run, cause some additional problems. At the other end of the spectrum are the xenomorphic agents. Newer entry, very exciting. They repair senescent cells. These repaired cells remain. They remain as non-dividing cells, but unlike the senolytic agents that remove the cells, the xenomorphic treated cells remain. Just like the senolytic agent therapies, they allow the surrounding cells to function more normally. Critically, they do not deplete the organ of the total number of cells. So this opens a different pathway possibly for repairing and reversing aging. So let's look at senolytics. Senescent cell count decreases because the cells are removed. They reduce the number of cells, restore function in many ways, can increase longevity, may increase health span. Most of the data is in mice. However, some of the exciting human data has been coming out of Mayo Clinic. Dr. James Kirkland and his team there have looked at quercetin, which is a plant polyphenol, and recall that these plant polyphenols, a lot of them are bright colored, high antioxidant, high activity dietary agents. We see them, for example, the Mediterranean diet. So these are around us and in our daily life. And combined with dasatinib, a leukemia drug, as oral therapy, they've been shown to have senolytic effects. There's a little study shows improved function in idiopathic pulmonary fibrosis. There's a great study out of Mayo with Dr. Kirkland's group looking now at what happens with systemic treatment for oral therapy. One of the diabetic kidney studies showed after only 3 days of oral therapy, so just a pulse therapy, there were reduced SASPs, inflammatory interleukin pathways were deactivated or reduced, and MMP degrading enzymes were reduced. So, and this continued after the oral therapy. So very interesting indeed. In contrast, xenomorphics also reduce the senescent cell count, but the cells remain. Professor Lorna Harries at Exeter has done some of the pioneering work. She's been using analogs of resveratrol, another substance we know from chocolate and red wine and grapes. And these small analogs were used to alter fibroblasts via the gene splicing. And you can see here a big decrease in senescent cells from the control to the treated. Fewer senescent cells, but no increase in apoptosis. That's important. That helps confirm that the cells are not leaving. So these splicing factor changes appear to cause true rejuvenation or repair and not senolysis and cell death. So another promising area for us. So in conclusion, both senolytic and xenomorphic therapeutic agents are promising. They promise improvement for health span, we hope, function of the body, and also improvement of the skin. So there are cosmetic aesthetic applications, and these could help us maintain youthful skin function and appearance. There's a potential role, particularly perhaps for the xenomorphics, for pre-rejuvenation, rejuvenation, even regeneration, and ongoing therapy, potentially as a daily or a pulsed routine. So we have a very bright future ahead, and some of the early data suggests that fat and human skin are affected by these senolytics as well. So thank you